Namaste, I am JVN. I am Dr. ANUPAMA Goyal. I am working as Associate Professor in Botany in Jyoti Vidyapeeth Women's University, Jaipur. Today I will deliver my lecture on transpiration, which is important for the graduate and undergraduate students of Jyoti Vidyapeeth as well as of other universities. Well, my topic is transpiration. So, what is transpiration? The loss of water in the vapor form from the exposed parts of a plant is called as transpiration. The loss of water due to transpiration is quite high. It is 2 liters per day in sunflower, 2 liters per day in sunflower, 36 to 45 liters in apple, and up to 1 ton per day in elm tree. So, this is the loss of water per day. Rather, 98 to 99% of the water absorbed by a plant is lost in transpiration. Hardly 0.2% is used in the process of photosynthesis, while the remaining is retained in the plant during growth. Now I talk about the types of transpiration. So most of the transpiration occurs through the foliar surface or through the surface of the leaves. It is known as foliar transpiration. So students, what is foliar transpiration? When the transpiration process occurs through the leaves, it is called as foliar transpiration. Foliar transpiration accounts for over 90% of the total transpiration. Young stems, flowers, fruits also transpire a lot. Mature stems transpire very little. So transpiration from a stem is called as choline transpiration. So what is choline transpiration? When transpiration occurs from the stem, it is called as choline transpiration. Depending upon the plant surface, transpiration is of the following 4 types: stomatal transpiration, cuticular transpiration, lenticular transpiration, and transpiration from bark. Now we talk about stomatal transpiration. So it is the most important type of transpiration. Stomatal transpiration constitutes about 50 to 97% of the total transpiration, and this stomatal transpiration occurs through the stomata. The stomata are found on the leaves. A few of the stomatas also occur on the young stems, flowers, and fruits. The stomata expose the wet interior of the plant to the atmosphere. Therefore, the internal air becomes saturated with water vapor. The outside air is seldom saturated with water except just after the rains. Water vapors therefore pass outwardly through stomata by diffusion. More water evaporates from the internal cells to replace the outgoing water vapors. The stomatal transpiration continues till the stomatas are kept open. Now see, this diagram is the vertical section of the leaf showing stomatal as well as cuticular transpiration. So this is the diagram of the leaf, and it shows the upper epidermis as well as the lower epidermis. What I mean to say, this upper epidermis and the lower epidermis, they are covered by a thick cuticle, and so the stomach transpiration process also occurs through the cuticle. Then it is called as cuticular transpiration. And now there are found stomata on the lower surface of the leaf, and when the transpiration process occurs through the stomata, it is called as stomatal transpiration. Now, this is the what is cuticular transpiration. Cuticular transpiration occurs through the cuticle or epidermal cells of the leaves and other exposed parts of the plant. In common land plants, cuticular transpiration is only 3 to 10% of the total transpiration. In herbaceous shade loving plants where the cuticle is very thin, the cuticle transpiration may be up to 50% of the total cuticular transpiration. Now I talk about the lenticular transpiration. So the lenticular transpiration is found only in the woody branches of the trees where lenticels occur. The lenticular transpiration is only 0.1% of the total transpiration. It continues day and night because lenticels have no mechanism of closure. So, lenticular transpiration continues throughout the day and night, and the lenticels connect the atmospheric air with the cortical tissue of the stem through the intercellular spaces present amongst the complementary cells. Now I talk about the bark transpiration. This type of transpiration means the bark transpiration occurs through the corky covering of the stems. Bark transpiration is very little, but it is measured rate is often more than lenticular transpiration due to the larger area. Like cuticular and lenticular types of transpiration, bark transpiration occurs continuously during the day and night. Now I talk about the mechanism of water loss in transpiration. In order to form vapors, water present inside the exposed parts of the plant requires a source of heat energy. It is the radiant energy during the day and heat energy from the transpiring organ during the night. In both the cases, the temperature of the transpiring organs comes to lie 2 to 5 degrees centigrade below that of the atmosphere. The atmosphere is rarely saturated with water vapors. The dry air of the atmosphere has a high DPD, diffusion pressure deficit, or low water potential up to 13.4 atmospheric pressure at 99% relative humidity. Such a high DPD means diffusion pressure deficit or low water potential can overcome various types of resistance water molecules have to meet in changing from liquid phase to vapor phase, and the movement of water vapors out of the transpiring organ. So the intercellular spaces of the transpiring organ is almost saturated with water vapors. When the stomata are open, the water vapors are drawn from the substomatal cavities to the outside air due to the high DPD, diffusion pressure deficit. And now I'll show you the mechanism of stomatal transpiration. The stomata, they function as Turgor operated valves because their opening and closing movement is controlled or governed by the turgor changes of the guard cells. Whenever the guard cells swell up due to the increased turgor, a pore is created in between them. With the loss of turgor, the stomatal pores are closed. Stomata open during the day and close during the night, with a few exceptions. The important factors which control or govern the stomatal opening are light, high pH or reduced carbon dioxide, and availability of water. The opposite factors govern stomatal closure, means stomatal closure is governed or controlled by darkness, low pH or high carbon dioxide, and dehydration. Now there are three main theories about the mechanism of stomatal movement, and these three theories about the mechanism of stomatal movement are very important from the examination point of view. So first of all, I talk about the hypothesis of guard cell photosynthesis. Well, as you all know, guard cells contain chloroplasts. During the day, the chloroplasts perform the function of photosynthesis and produce sugar. So sugar increases the osmotic concentration of the guard cell. Sugar increases the osmotic concentration of the guard cells, and it causes absorption of water from nearby epidermal cells. So the turgid guard cells bend outwardly and create a pore in between. However, the photosynthetic activity of the guard cell chloroplast seems to be negligible. Now I talk about the classical starch hydrolysis theory. Now the main features of classical starch hydrolysis theory were first of all spelled out or proposed by Sayer in 1923. Then it was modified further by Stewart in 1964. The guard cells contain starch at low carbon dioxide concentration in the morning achieved through photosynthesis by mesophyll and guard cells. pH of the guard cells rises. What I mean to say, the guard cells contain starch, so at low CO2 carbon dioxide concentration, pH of the guard cells rises or increases, and it stimulates enzyme phosphorylase. It stimulates the enzyme phosphorylase. And now this phosphorylase enzyme converts starch into glucose 1-phosphate. So the glucose 1-phosphate is changed to glucose 6-phosphate, which undergoes hydrolysis to produce glucose and phosphoric acid. Now you see in this diagram, or the material shows the starch under the presence of phosphorylase enzyme is converted into glucose 1-phosphate. Now this glucose 1-phosphate in the presence of the enzyme phosphoglucomutase is converted into glucose 6-phosphate, and glucose 6-phosphate with the help of water and phosphatase enzyme again gets converted into glucose and phosphoric acid. This I mean to say, and now I show you glucose increases the osmotic concentration of the guard cells. On account of it, the guard cells absorb water from the neighboring cells and swell up and create a pore in between them. And in the evening, there is a closure of the stomata. So evening closure of stomata is brought about by the increased carbon dioxide content due to the stoppage of photosynthesis of leaf. It decreases the pH of the guard cells and brings about phosphorylation of glucose in the presence of the enzyme phosphorylase. Glucose 1 phosphate is changed into starch. So this equation shows glucose in the presence of ATP and hexokinase enzyme is converted into glucose 6 phosphate and ADP. And now this glucose 6 phosphate in the presence of the enzyme phosphoglucomutase is converted into glucose 1 phosphate, and now this glucose 1 phosphate in the presence of the enzyme phosphatase is converted into starch and phosphoric acid. Now, as a result, osmotic concentration of the guard cells falls. The guard cells lose water to the adjacent epidermal cells. With the loss of turgidity of the guard cells, the guard cells shrink and close the pore in between them. Now I tell you what were the objections raised for this theory. First, glucose is not found in the guard cells at the time of stomatal opening. Second objection was starch sugar interconversion changes. Starch sugar changes are chemically slow, while opening and closing of the stomata are quite rapid. Third objection was wide changes in pH of the guard cells cannot be explained on the basis of carbon dioxide concentration. Then the fourth objection was onion and some of its relatives or species do not possess starch or polysaccharides that can be hydrolyzed to the level of glucose. Fifth objection, blue light has been found to be more effective than any other wavelengths for the opening of stomata. The same cannot be explained by starch hydrolysis theory. Sixth objection, hydrolysis of starch theory cannot account for high rise in osmotic pressure found in the guard cells. Now, this diagram shows the conversion of starch into glucose phosphate. First, see this starch is converted into in the presence of the enzyme phosphorylase. Starch in the presence of the enzyme phosphorylase is converted into glucose 1 phosphate having the low osmotic pressure, and now this glucose 1 phosphate is converted into glucose 6 phosphate under the influence of the enzyme phosphoglucomutase, and now this glucose 6 phosphate is converted into glucose plus phosphate under the influence of the enzyme phosphatase with the release of ADP, and ATP is converted into ADP. Now, next theory is malate or potassium ion pump hypothesis. This theory is the modern theory, and now the main features of the malate or potassium ion pump hypothesis were put forward by Leavitt. Means this theory was proposed by Leavitt in 1974. According to this theory, pH of the guard cell can rise due to the active hydrogen uptake by the guard cell chloroplast or mitochondria, CO assimilation by mesophyll and guard cells. A rise in pH causes hydrolysis of starch to form organic acids. Means when the pH of the guard cells rises, there is the hydrolysis of the starch into organic acid, especially phosphoenol pyruvate. Starch is converted into hexose and hexose phosphate, and hexose phosphate is again converted into phosphoenol pyruvate. Phosphoenol pyruvate can also be formed by pyruvic acid of respiratory pathway with the help of the enzyme PEP carboxylase. PEP carboxylase, it combines with the available carbon dioxide to produce oxalic acid, which gets changed into malic acid. Malic acid now dissociates into hydrogen ion and malate ion. So the hydrogen ions pass out of the guard cells actively. What happens? Malic acid dissociates into hydrogen ions and malate ions. Now the hydrogen ions pass out of the guard cells. In exchange, potassium ions, they pass inwardly, and so similarly the chloride ions may also enter the guard cells along with the potassium ions. So the potassium ions are entering inside the guard cells, and now the guard cells maintain their electroneutrality by balancing the potassium ions with malate and chloride ion. So in the combined state, they pass into the small vacuoles and increase the osmotic concentration of the guard cells. As a result, guard cells absorb water from the nearby epidermal cells through endosmosis, swell up and create a pore in between them. During stomatal closure, the hydrogen ions diffuse out of the guard cell chloroplast. It decreases the pH of the guard cell cytoplasm. Any malate present in the cytoplasm combines with hydrogen ion to form malic acid. So the excess of malic acid inhibits its own biosynthesis, and high carbon dioxide concentration also has a similar effect. So the undissociated malic acid promotes leakage of the ions. As a result, potassium ions dissociate from malate and pass out of the guard cells. Now the next one, what are the factors which affect stomatal movement? So there are certain factors which affect the stomatal movement: light, temperature. So I will talk one by one. The factors affecting stomatal movements. Stomatal movements are influenced by a number of environmental factors like light, temperature, humidity, water availability, and CO2 concentration. Thank you.